Hey everyone, so I'm going to be discussing Lab 21 on cardiovascular physiology. I'll go over a demonstration of the microcirculation within a goldfish tail, as well as how to do 21B, taking a venous pressure, and then of course how we measure arterial blood pressure with a blood pressure cuff. So in lab, we usually do a goldfish demo. We look at the circulation of the blood in a goldfish tail. So normally we would anesthetize the goldfish. We place a type of anesthesia in the water that the goldfish is swimming around. And once the goldfish has been anesthetized, we then place them in a little petri or culture dish. And then we'll have the fishtail kind of sitting on a stack of microscope slides. And then we get to observe the circulation of blood within the goldfish tail. So this is sort of a couple pictures of what essentially what you would be seeing. Normally the blood cells would not be colored like this. So in the goldfish tail you'd be able to see veins and arteries. They wouldn't be colored as it is in this picture, but you would see structures, you'd see outline of structures. So the bamboo that you see in the picture, so this is what the pictures that you would see under the microscope. The bamboo-like structures, that actually, those are the cartilage that you'll see within the tail. What we want to look at are the muscles between the cartilage in the tail, and that's where you'll see the blood vessels, and that's where the blood vessels would be located. Now you can search for goldfish circulation videos on YouTube on your own, but I have actually included a video here so that we can see for ourselves. So some of these videos actually do have music accompanying the video. What I'm going to do is kind of bring that down. There you go. So as you can see the cells kind of circulating within the blood vessels located within the goldfish tail. It actually looks really cool. The flow of blood and the blood cells there. Just to observe the circulation of blood in the arteries and veins of the goldfish. If you kind of look closely, you can see the different cells and the cell structures, the biconcave shapes of the erythrocytes. So I will leave that up there. I'm not going to play the whole entire minute and a half of that, but you get the idea of the circulation that can be observed within the goldfish tail. The next part of the lab is part B, which is measuring venous pressure. When we talk about blood pressure, we're actually talking about arterial blood pressure. Basically, it's an estimate of the blood pressure within the aorta. This part of the lab is actually going to kind of sort of measure venous pressure, or the pressure within the veins. So we know that within the body, fluids move due to pressure differences, and they will move from, a high pressure to a low pressure. So within our body, the highest pressure that we have will be in the left ventricle, because the left ventricle has to have enough pressure to push blood out of the heart to the rest of the body. So the left ventricle will push blood towards the arteries, then the capillaries, the veins, and then back to the right atrium. What we want to measure, there has to be a large amount of pressure within the veins in order for the blood to move from the veins back into the atria. So we kind of want to have an idea of what that pressure is. So for this experiment, you would have a person standing next to a wall or the chalkboard, so have their shoulder kind of standing right next to the wall. And then you want to mark the height of the atria, specifically the right atrium on the wall. Now, this is around the area or the height of the armpit, about the anterior axillary line. So you would use a ruler or a yardstick to kind of measure that line. And then at that point, you would have your volunteer hold their hand at the side below the heart, where the blood will pool in the hand due to gravity, and the veins in the hand will swell or distend. Then have your volunteer raise the hand slowly. Once it gets above the heart, the gravity will actually allow for venous flow from the veins in the hand, and the veins will collapse when the pressure within the vein is greater than the pressure in the atria. So I actually do have a video showing you how to do that by my lovely assistant. So hopefully you can hear this. Hey everyone, so I'm going to show you how to do the first part of Lab 21, finding out about venous pressure. So I have my lovely assistant Erin, who's going to help me show you how to do this part of the lab. So according to the lab, you want to measure where exactly the atria would be. So that would be around the area of the anterior axillary line. And I've marked that on the wall, on a piece of paper, of course, with a line. You're then going to have your arm just by your side so that the blood in your hand pools in the hand. You're then going to raise your arm. So Erin, raise your arm until you see, right about there, until you see that the veins in the hand have returned back to normal, showing that the blood that pooled in the veins in the hand has returned back to the heart. And when you see that the veins have sort of collapsed, this is where you want to measure your arm on the wall. So you can take a ruler and then place the ruler exactly where the veins have collapsed, and then you measure that in millimeters. Okay, so I'm going to pause this while I mark it on the wall. So as you can see, I placed a mark on the wall around the area where the veins collapsed after you raised the arm. So Erin, you can put your arm back down. And then you're going to measure the distance between the two lines, and then make sure the measurement is using millimeters. I was able to measure the distance using a ruler, and with the help of my lovely assistant Erin. The distance between the two lines was about 95 millimeters. So you would take this number and then put it into a formula. With regards to venous pressure, so the measured distance from the height of the right atrium to where the vein collapsed for my volunteer was about 95 millimeters. This will correspond to a venous pressure. So basically what we would do is we would divide this number of millimeters by 13.6. Now, where does 13.6 come from? 13.6 is actually the difference between the density of mercury, because back then we measured things in millimeters mercury. So it's the difference between the density of mercury and the density of blood. So if we take 95 millimeters and then divide that by 13.6, I have a number roughly about 7. So using that formula here, so pressure in the venous pressure equals vein collapse minus right atrium. So that height we said was 95 millimeters, divided that by 13.6. The number that I got was about 7 millimeters mercury. So normal range is usually between 4 to 10 millimeters mercury. Now, this isn't a precise number, of course. If you really wanted to find out what your venous pressure was, you'd have to go to a lab and have that measured. And a lot of things could be taken into consideration. For example, sometimes we do overestimate the height of the of where the vein collapses. Also, we can overestimate the height of the right atrium. So normally these numbers are a lot bigger, and you'll see bigger numbers in lab. So it's not perfect, but this is the formula that we use in lab. So basically the difference in height between where the vein collapses in the hand as well as the right atrium, and then divide that by 13.6. The next part of our lab is measuring arterial blood pressure. In systemic circulation, the highest pressure occurs in the aorta and results from pressure created by the left ventricle during ventricular contraction. Aortic pressure can reach an average high of about 120 millimeters mercury during ventricular systole. This is the period of ventricular contraction, at which point this is known as our systolic pressure or systole. Pressure will then fall steadily to a low of around 80 millimeters mercury during ventricular diastole, also known as ventricular relaxation or ventricular filling when the ventricles fill with blood. This is diastolic pressure or diastole. So this part of the lab is when we are actually measuring blood pressure with each other. So clinically, we use a fancy instrument called a sphygmomanometer. I actually use that word for spelling, and my students have to know how to spell sphygmomanometer, let alone, be able to say it. So we use a sphygmomanometer, just a fancy word for a blood pressure cuff, to measure systolic pressure and diastolic pressure. So the blood pressure cuff is used to occlude the brachial artery, that main artery within the arm. We occlude the brachial artery at a certain pressure above normal systolic pressure, and then once we've occluded that artery, we then slowly release the pressure within the blood pressure cuff until we hear audible sounds. These sounds are called carot cough sounds, and we use these sounds to help us measure systolic and diastolic pressure. So the first sound we hear is the blood being let back into the brachial artery. That first thump that we hear, that is an indicator of our systolic pressure, and we keep releasing the pressure within the blood pressure cuff until we hear the sounds stop or disappear, and that would be your second indicator for the diastolic pressure. So once the sounds stop, that's where you read the gauge, and then that would be an indicator of your diastolic pressure. So this is how we would measure systolic and diastolic pressure in the lab. You place a sphygmomanometer as well as a stethoscope on a partner's left arm. So what you want to do is place the cuff about 2 to 3 centimeters above this cubital fossa in the elbow, and then you want to follow the artery medially, the ulnar artery, up until it is coming from the brachial artery medially. So you want to place the stethoscope just near the brachial artery. Before all this, of course, make sure your cuff has been fully deflated. I tell my students to make sure they fully deflate the cuff before placing it around the patient's arm. You also, before you take a measurement, there are questions you need to ask. You have to ask them whether or not they've done any exercise, smoked any cigarettes, or drank any coffee or caffeinated drinks, because these could affect blood pressure. And you need to make note of that in the patient's chart, whether or not they've done any of these things, in case their blood pressure is unusually high. So once you've placed the stethoscope as well as the sphygmomanometer on the person's arm, you then have the patient relax their arm. I like to make sure the patient's feet are both on the ground, and then you use the bulb to increase the pressure in the cuff. Now, it says here, pump up the cuff to 160 millimeters mercury, no higher. However, if you have a patient that has a history of high blood pressure, you want to go a little bit higher than what their normal is. And usually they will know what their normal blood pressure is, or you can check their chart. So you occlude the brachial artery, and then you will slowly release the pressure in the sphygmomanometer, and using a stethoscope you will listen for your carot cough sounds. So systolic blood pressure or systolic pressure is indicated by that first swooshing sound, that first carot cough sound. That is where blood is initially entering the occluded brachial artery. So systolic pressure is that first carot cough sound. You then continue to listen as you continue to release the pressure in the sphygmomanometer until finally when no more sounds are heard, at which point you will record the diastolic pressure. It takes a little bit of practice to get the hang of listening for the carot cough sounds. One lab I was fortunate they had double stethoscopes, so I could help the student listen for the carot cough sounds. But once you keep practicing, you kind of get the hang of it. So again, you use a sphygmomanometer or a blood pressure cuff to measure systolic as well as diastolic pressure. So we increase the pressure within the sphygmomanometer until we completely occlude the brachial artery. So if there's no sound, that means the cuff pressure is above systolic pressure and the artery is completely occluded during the cycle. As soon as the cuff pressure is just below systolic pressure, blood is allowed to re-enter the brachial artery, and you'll hear that first sound. It's a soft tapping sound, and it's intermittent. That is your carot cough sound. That is where you will measure systolic pressure. And then you keep allowing more, you keep releasing the pressure in the blood pressure cuff slowly, and you'll keep hearing these intermittent sounds and tappings, this carot cough sounds, until finally the brachial artery is free to flow, and then you will no longer hear the sounds. As soon as you no longer hear those carot cough sounds, that is where you will measure your diastolic pressure. So the cuff pressure is below diastolic pressure, so the vessel is always open, and then there's no more turbulence and no more sound. So the sounds you're hearing are basically blood being allowed back into that artery. So the lab that you're supposed to do, you're supposed to take blood pressures among your fellow classmates, and then you want to calculate these values. Let me go into a little bit of explanation of these values. First, let's talk about pulse. So pulse is the rapid pressure increase that occurs when the left ventricle pushes blood into the aorta, transmitted through the fluid-filled arteries, and this pulse is also known as a pressure wave. Now, the pulse pressure is a measure of the strength of this pressure wave, or the measure of the strength of the pulse, and we get this number by using this formula. So pulse pressure, or PP, equals your systolic pressure minus the diastolic pressure. So again, this measures the strength of the pulse, or measures the strength of the pressure wave. We know that arterial pressure, or blood pressure, reflects the driving pressure created by the pumping action of the heart. Now, while it is difficult to measure ventricular pressure directly, we assume that arterial pressure reflects ventricular pressure. So when we're trying to find mean arterial pressure actually represents this driving pressure. And there are different ways that we can get mean arterial pressure. So we can use the formula 1/3 of the systolic pressure plus 2/3 of the diastolic pressure. Now, it's a lot of math and it's a little more complicated. However, if we use this formula, taking the diastolic pressure plus 1/3 times the difference between systolic pressure and diastolic pressure, or the pulse pressure, it's a little bit easier to get that number. So I recommend using this part of the formula, so diastolic pressure plus 1/3 times the pulse pressure, and it's a little bit easier to calculate the mean arterial pressure. So during lab, we take a volunteer and take 3 measurements, 3 blood pressure measurements, and then put them in a table. Again, we can find pulse pressure using that formula, finding the difference between systolic pressure and diastolic pressure, and then using the values that we obtain, we can find the mean arterial pressure using either one of these formulas for mean arterial pressure. So the experiment that you're supposed to do in lab, or that we were supposed to do in lab, if we had had access to sphygmomanometers and stethoscopes, you wanted to be able to determine mean arterial pressure while having your volunteer either sit for 2 minutes, and then you take blood pressure immediately after standing, and then standing after 2 minutes, and then taking up pressure after reclining supine for 2 minutes. So we get the blood pressures, the arterial blood pressures, and then we take those values and obtain the mean arterial blood pressure for these different instances. So that would be that lab. This is the end of the explanation of the lab. There will be a separate video for the discussion on the cardiovascular physiology. In the meantime, there is a lab handout that has values where you can do the formulas for mean arterial pressure. That will be posted with this video, and that handout will be due on Friday.